Hello guys, I'm Bongani. Welcome to my YouTube channel. And guys, please don't forget to subscribe to my channel. Don't forget to like, don't forget to share, and of course, comment down below. And if you have any question, please, I'd be happy to actually receive your message on the comment section down below. So guys, today on our lesson plan, we are going to look at nucleic acids. First of all, I want us to look at our lesson plan, or I want us to look at what we are going to discuss today. First of all, I want us to actually revise or do revision on the structure of a cell, because there's a very important part of a cell where these nucleic acids are located. We need to know that specific part. Hence, I want us to go back to grade 10 work or content. And we are going to also look at the brief history of the discovery of the DNA molecule by Watson and Crick and Franklin and Wilkins. And then we're going to look at the two types of nucleic acids, which are your DNA and your RNA. And for DNA, we're going to look at the location or where it's found. We're going to look at its structure. And of course, we also need to look at its functions. And also, again, for DNA, we are going to discuss DNA replication and DNA profiling. And last but not least, we are going to look at the other nucleic acid known as RNA, which also we're going to look at its location, we're going to look at its structure, and of course, its function. Now, looking at the structure of a cell, you all know these other structures, you all know them, your vacuole, your cell membrane, you all know those things. We studied in grade 10. Okay, now, what is important for you to know here is this part here, the nucleus. I want to emphasize that it's very important for you guys to know the nucleus, especially when we talk about DNA. It's very important, it's very, very important. And also, we're going to do also protein synthesis, but not on this lesson, on another different lesson. So it's very also important for you. It's also important to know these tiny dots, these tiny structures, your ribosome, and of course your cytoplasm. Okay, now I want to dwell much on the structure of a cell because we all did this in grade 10. It's grade 10's work. Now moving right along, we are going to look at the discovery of the DNA molecule. Or we're going to look at how, when, and who discovered what? The structure of the DNA. Okay, now. In early 1950s, Maurice Wilkins and Rosalind Franklin, a physicist and a chemist respectively, showed by using X-ray diffraction or X-ray image to show us a pattern of regularly repeating nucleotides, and this was the first clue to the three-dimensional structure of a DNA molecule. Now, we are going to look at the structure of the nucleotide later and its components. We're going to look at that later on our lesson. But now, what is important is for you to understand that these two-faced scientists in the early 1950s, what they discovered is that the DNA molecule consists of a chain of subunits called nucleotides. Okay, that's what they discovered then. Okay, and then finally in 1953, James Watson an American biochemist, and Francis Crick, an English physicist, began their collaborative work to try to solve the puzzle of the molecular structure of DNA. So using data provided by Maurice Wilkins and Rosalind Franklin, they made an accurate model of the molecular structure of DNA. So this discovery, they called it the secret of life. So guys, here what happened is that later, after Maurice Wilkins and Rosalind Franklin discovered that the structure of DNA is consists of or is comprised of a chain of nucleotides. So these other scientists, Watson and Crick, they actually brought together the data that they got from these two other scientists to actually make an accurate model of how a DNA molecule look like. Okay, and then in 1962, Crick, Watson, and Wilkins, they all received the Nobel Prize for determining the molecular structure of DNA. And if you have noticed, it's only three of them who received the Nobel Prize. What about Rosalind Franklin? Guys, Rosalind Franklin, unfortunately, uh, she died. It's a lady. She died, and Nobel Prizes cannot be given to people who actually have already passed on, unfortunately. Hence, she was not actually honored for actually playing a pivotal role for a pivotal role in actually discovering the model of how the DNA looks like. So what did the scientists find is that the DNA is a double-stranded molecule and has a helical shape, or it's a double helix, meaning that it is a spiral. Yes, it's a spiral form of a molecule consisting of two DNA strands which are wound around each other. Okay. And then they also discovered that it is complementary, meaning these two chains or these two strands, they complement one another. But we're gonna look at that. We're gonna look at that when we discuss nitrogenous bases, the complementary pairs of nitrogenous bases. They complement each other, meaning one has to bond with the other one has to bond with the other one through some particular bonds, which we're gonna discuss. And what also they discovered is that this molecule is anti-parallel. What does that mean? It means now that it is composed of two strands that run alongside each other but point in different directions. Okay, now let us just continue with our lesson. Now, looking at the structure of DNA, guys, DNA in full is called deoxyribonucleic acid, but it is abbreviated as DNA to make things easy. You won't be penalized on your exam if you write just DNA, because I feel like it's better for you to write it as DNA so that you do not actually put the wrong spelling, because you might spell it incorrectly and then it sounds like something else that we do not know. And by doing that, you will lose points or you will lose marks. So I prefer that students they write it as DNA. Okay, to avoid losing marks. So looking at DNA. So DNA, as we all know now, it's a nucleic acid, since I told you that today we're going to do nucleic acids. And then this nucleic acid is made up of two strands and it is located in the nucleus. Hence, I wanted us guys to look at the structure of a cell, particularly the nucleus. That is where we find our DNA. But guys, DNA is not only found in the nucleus. We have another DNA we call mDNA, which is messenger DNA. But we're not going to dwell much on that. It's not important at your level. It's not really important. And it is these two strands, they are wound around one another to form a double helix, which is what we talked about when we were talking about the discovery of the DNA molecule structure. And then each DNA strand is made up of nucleotides. And here's something also I told you about, that it was discovered by Wilkins and Franklin. Okay. And then each DNA nucleotide consists of a deoxyribose sugar. It consists of the phosphate group. It consists of 4 nitrogenous bases, which are your adenine, your thymine, your guanine, and your cytosine. Okay, guys, don't be confused by this. I'm going to discuss how does a nucleotide looks like. Okay, now the two strands are connected by what we call weak hydrogen bonds. So guys, it's very important for you to emphasize that these bonds are weak, because if you don't do so, you're going to forfeit points. Okay, but sometimes the people who mark your scripts during matric, they can be lenient sometimes. They accept hydrogen bonds. But guys, to avoid losing marks, please include the weak. Okay, they are weak hydrogen bonds because they have to be broken down, during a particular process. Now, these bases, they just don't bond with any base, but now a specific nitrogenous base will bond with a specific nitrogenous base. So adenine always bonds with thymine, okay? And then guanine always bonds with cytosine. Now, let us just look at the components of a nucleotide. Okay, looking at the structure of a nucleotide, guys, as I told you, as you can see here, now guys, a nucleotide is not only found on DNA, it's found also on RNA. Okay, now a nucleotide, guys, it's this structure here on your left, on your screen, this one, the one I'm pointing at. Okay, so this structure, it consists of 3 main components, important main components, because these components can be asked by your examiner. So you need to know how to label them. Okay, now we have the phosphate group. Sometimes some people call it the phosphate, it's okay. And we've got the sugar, and of course, we've got the nitrogenous bases. Okay, but now guys, we have 2 types of these sugars found on nucleotides of these 2 nucleic acids. So for DNA, we have a specific sugar. For RNA, which is what we're gonna discuss later, has its own specific sugar. So at this point, because we talked about DNA and we're still gonna talk about DNA. So DNA, its sugar is called the deoxyribose sugar. Okay, but now for RNA, the sugar is called the ribose sugar. Okay. Also, these nucleosides, they are classified, into 2 different classes. We have got the purines, which is your adenine and your guanine. And then we've got your pyrimidines, which are your cytosine and your thymine. So guys, now looking at the structure of our DNA, remember guys, our DNA is a double-stranded molecule, or it's a double helix. Okay, so this chain here on your left, it's a chain of nucleotides bonded to one another, and then it's also found on your right. So it's a double what? Stranded. It has 2 strands. It's a double-stranded molecule. Now, in each and every nucleotide, as I've discussed, we have got a phosphate, we have got a sugar, and one of the 4 nitrogenous bases. Okay, they are joined together. As you can see here, they are bonded together. Now, between 2 complementary nucleotides, sorry, nitrogenous bases, we have what we call your weak hydrogen bonds. These bonds here in between these complementary nitrogenous base are called what? They are called the weak hydrogen bonds. And also what I want you guys to also bear in mind, or something that you must know, is that between your adenine and your thymine, we only find two weak hydrogen bonds. But for guanine and cytosine, we find three bonds of weak hydrogen bonds. Okay, let's continue. That is not important, but I want you to know that, because some examiners, they can be crazy and ask you this, but at your level, it's not important to know how many bonds between each complementary base pairs. But these examiners can be crazy sometimes. What is important for you to know is that the bonds are called weak hydrogen bonds. Now, let us just look at the location and the functions of DNA. Okay, now you have got DNA that is found within the nucleus, which we call the nuclear DNA, which makes up the genes on chromosomes. Okay, and then also we have got the mitochondrial DNA, which is found in your mitochondria. Okay, now looking at the functions of DNA, we have got two functions, which is what you're supposed to know at your level. Okay, what is important is you to know that the sections of DNA forming genes carry what? Hereditary information. So your DNA has got what? Genetic material. Okay. And also DNA contains coded information for protein synthesis. That's all that you need to know at your level. At this point, you need to know these two functions. It carries what? Genetic information. Okay, and also it contains coded information for protein synthesis, because DNA is used during protein synthesis to form mRNA, which is something we're going to discuss when we do protein synthesis. At this point, you don't need to understand it, but we'll understand it later when we do protein synthesis. Now, I want us to look at this very important process that occurs in an animal cell. Okay, it's very important for you to know this process, because you might be examined on this process. It's very important for you metics to understand the process, the entire process, because sometimes they might give you an entire essay on this topic on DNA replication. So it's very important for you to know it. So DNA replication takes place at the interface before mitosis or meiosis begins. Okay, so we're all gonna look, okay, because I haven't done meiosis and mitosis, but I'm gonna have a lesson on that. You're gonna understand that when we do mitosis and meiosis. Now, it happens during a phase called interphase before mitosis or meiosis. And then the DNA replication is the process during which a DNA molecule makes an exact copy of itself, meaning that DNA actually photocopies itself. It makes copies of itself. So it replicates. It forms a replica or a duplicate, another image of itself. Okay, it's very important for you to understand that. So now, there are some certain steps that occur during DNA replication. You need to know these steps by heart, and they're not very difficult. And actually, I've highlighted the most important parts of the steps that are very important, that will give you marks if maybe you're asked to explain the process in an essay. Okay, now, because the DNA, as you can see here, the strands are wound around each other. Okay, now the first thing that has to happen is for the structure to unwind. So the double helix unwinds. You see here, the structure unwinds. Okay, it opens up. Okay, now what happens is that hydrogen bonds, the weak hydrogen bonds, guys. I'm going to emphasize that again. The weak hydrogen bonds between the nitrogenous bases, I made a mistake, they're not hydrogenous bases, they are nitrogenous bases. They break, okay? Because they're weak, so they break. And two DNA strands unzip or they separate. Guys, after these bonds here have been broken, what happens is that now these two strands, they're starting to move apart from each other. They separate or they unzip. That's a very important point. They separate or unzip. They move away from each other. They are torn apart. And then each original DNA strand is used as a template. Okay. Now, what happens is that free nucleotides build a DNA strand onto each of the original two strands by attaching their complementary nitrogenous bases, where adenine bonds to thymine and guanine connects to cytosine. Okay, now these act as a template. Each original strand act as a template. Okay, these two original templates, they are the ones that they're going to be used, to form more copies. Okay, now what happens is that because now we have got free nucleotides in our cell, so these nucleotides, they use these templates to form a new DNA molecule, a double-stranded DNA molecule. So what happens is that now the complementary nitrogenous bases, they bond, whereby now adenine goes with thymine and cytosine goes with guanine. And then once that happens, how do they bond? They bond through what? Weak hydrogen bonds. The hydrogen bonds now are formed again. The weak hydrogen bonds are formed again. And then once that happens, two identical DNA molecules are formed. Okay, and then each molecule consists of one original strand, which was the template, and a new strand, which is the free nucleotide. I want you guys to understand that it's very important. It's not that difficult. It's very easy. What happens? The double helix unwinds, which is what you see here. Number one, it unwinds. Very important. Then once the double helix unwinds, what happens? The weak hydrogen bonds, they break. Once they break, the structure starts to unzip, or the two strands, they move apart from each other and they're separated or they're unzipped. And then after that, each template is going to be used to form other copies, whereby now new complementary nitrogenous spaces, they're going to bond or they're going to connect to other nitrogenous spaces according to the rule of the complementary rule that adenine bonds with thymine, cytosine with guanine, just like that. And two identical DNA molecules are formed. Okay, and then each new DNA molecule is formed consists of the original part of the template of the double helix and the new one from the free nucleotides in the cell. Now, I want us to also discuss the significance of DNA replication. We have talked about this process, but now, why is it important? It does not just happen without any reason. It has to happen because of some certain reasons. It does not just happen. Okay. Now, what happens is that this DNA molecule, DNA replication, because now new copies are formed, or new replica is formed, or new duplicates are formed. What happens? It doubles the genetic material or information, so it can be shared between the resulting daughter cells during cell division, during meiosis and mitosis, which is what we're going to discuss. But for you now, what you need to know is that it doubles the genetic material so that it can be shared between the resulting daughter cells during cell division, when your cells divide. And also, it results in the formation of identical daughter cells during mitosis. Remember, okay, because we haven't discussed mitosis, but briefly, mitosis is whereby now the cell divides, to form identical daughter cells. But we're gonna discuss that when we do mitosis or cell division. Okay. Now, let us just look at DNA profiling. DNA profiling is a very important topic because this thing, it helps, especially when it comes to crime. It helps a lot, and it also helps when some fathers do not want to who, like those fathers who deny their kids. Guys, this is really a very important procedure that is done by scientists, to actually solve cases of crime. Now, what is DNA profiling? DNA profiling is the process whereby a specific DNA pattern called the profile is obtained from a person or a sample of a bodily tissue. Example, your nails, your hair, your blood. So here, they want to find a certain DNA pattern for a particular person. Because remember, each and every person has a different or has a unique DNA. My DNA is not similar my to my brother's DNA, even though we are one. We are from the same parents, but that doesn't mean we have the same DNA. So I've got my own code of life. My other brother has his own code of life. Okay, we're not the same in terms of the DNA. My DNA pattern is not as the same as your DNA pattern. We have different DNA patterns. Okay, although we can be from the same parents, but they are not the same. Now, this DNA pattern can be obtained from each and every person, and it can be obtained from certain fluids in our bodies or from certain tissues in our bodies. So it can actually be found from your nails, under your nails or your nails actually, and then it can be found your hair. Your hair contains your DNA, your pattern of DNA. It can be found from blood. It can be found from semen. It can be found from your clothes, basically. Your dead skin cells, from your skin. Okay, that pattern is very important. I'm gonna tell you why it's so important. Why is it so important? But now we also need to define what a DNA profile is, because there's a huge difference between DNA profiling and DNA profile. So I want you to understand these two different terminologies, because in an exam you might be asked to actually give a definition of DNA profiling or a definition of a DNA profile. So a DNA profile is actually the result of DNA profiling. So it's a barcode, a pattern formed from DNA that is unique to each person. It's not a profile. It's like the it's like a paper that shows a certain DNA pattern of a person, of a particular person. Yeah. And then it is used actually in crime scenes. How? Now we're going to look at these uses of DNA profile. Guys, I love DNA profiling because it's really played a pivotal role in our society, because you find cruel people who just get away with murder, and you find fathers who do not want to maintain their kids because they're denying their kids. So this actually brought some order in the society, and I like it because most kids now, they're able to know who their fathers are, because fathers used to deny their kids, even though some people still continue, but some people need closure to know who their fathers are. Okay. Now, how can DNA profiles be used? So they're used to identify criminals. For instance, for the example of the DNA profile that I gave you guys on your screen, this one on your left is actually a DNA profile that was found from the crime scene. And now people were arrested, those were the suspects, the suspected ones who would have just took place in a particular crime. Okay, now they collected their DNA profiles. Now, because now we want to know who actually did the crime, you know, who exactly participated in doing crime. We want to know the person. Maybe, for instance, somebody was killed at their place. Now we need to actually research, do your work. Your detectives now, they're doing their research to find who actually murdered the person. Now, they take a particular DNA sample found from the crime scene, and then they take it to lab to get the DNA profile. And then now, they arrest the people actually who are suspects, and they take their sample as well of DNA, and then they get their profiles as well. And then now after that, they compare. Okay, once they compare, if a certain suspect's DNA profile looks exactly like the DNA pattern that was found on the crime scene, that's a problem, actually. Now, the person has to be arrested. You know, the person has to be arrested because clearly it has been proven that that particular suspect is the one who actually committed crime, is the one who actually did murder the person. That person has to be what? Has to be taken to jail, and like that person has to be given a particular sentence by the court and so forth. I'm not a lawyer, I don't know those things. So guys, now looking at our example, usually they like giving you crime scenes, or they can give you ask you who's the father of the child. Do you get me? And then they're gonna give you DNA, who possibly the father can be. But now you want to compare who's the exact father, the biological father. Because even though, guys, remember I said our DNAs are not the same, but now because we are related, it has to be the same some way. It won't be completely the same, but they'll ask you who is possibly the father. Hence, sometimes DNA comes says 90%, you are 90% the father. So the DNA results came back positive, meaning you are the father. Okay, such cases. Yeah. So for a crime scene, they can give you the DNA sample or DNA pattern or profile that was discovered from the crime scene, the place where the crime took place. And then they're going to show you the DNA of the suspects, suspect 1, 2, 3, or suspect A, B, C, or they can give actual names. Okay. Now they'll ask you who is actually the one who committed the crime. So what you need to do, you need to look at these bars here, this barcode, the entire barcode. Now, as you can see here, suspect number 2 is the one who committed the crime. So that's the person that has to be arrested by the police, because looking at the DNA profile found on the crime scene is similar to the suspect's DNA that was found from the sample from the suspect. Okay, look at the bars, exactly the same. 1, 2, 3, 4, 5, 6. So it's 1, 2, 3, 4, 5, 6. You see? And also small bars, a bigger bar, small. Yeah, small, a bigger bar, small. So this is actually the person who committed the crime. Okay, now moving to RNA, guys. Now, RNA is similar to DNA somewhere, but it's not similar to DNA somewhere. So, guys, RNA in full is called the ribonucleic acid. And remember, when we were talking about the structure of a nucleotide or the components of a nucleotide, we spoke about that a specific nucleic acid has a specific sugar on its nucleotide structure. So for RNA, guys, we call it the ribose sugar. The sugar found there is the ribose sugar. This part here, this is a ribose sugar. Now, the location of the RNA. So RNA, mRNA, which is what we call the messenger RNA, is formed in the nucleus and it functions on the ribosome. So we've got RNA that is formed in the nucleus, which means it's located there in the nucleus, mRNA. And then also got we've got tRNA. tRNA is located in the cytoplasm. In full is what we call the transport RNA. mRNA, remember, messenger RNA, and then tRNA is transport RNA. We're going to talk about those when we do protein synthesis. Now, the structure of the RNA molecule. So guys, RNA is different in its structure compared to the DNA molecule. How so? You're gonna understand as I'm going to explain to you. Now, as it is a single-stranded molecule consisting of nucleotides. So this one is not similar to DNA because it is single-stranded. It consists of only one strand, as you can see, which is one chain of nucleotides joined together. Okay, and then each nucleotide is made up of a sugar called a ribose sugar, a phosphate, and a nitrogenous base. So now the components or the parts of the nucleotides here, the sugar is not the same. So the phosphate is the same except for the sugar. The sugar is not the same as the ribose. And the nitrogenous base for this one is the same, but there's a different one. Now, for nitrogenous bases of RNA, they are the adenine, which we found in DNA. We've got uracil, which is something new that we do not know of. So this one is a different nitrogenous base. The others are the same cytosine, and of course, guanine. So we don't have thymine here. So RNA consists of 4 of the nitrogenous bases: your adenine, your guanine, your uracil, and of course your cytosine. Now, the only function of RNA is that it plays a role in protein synthesis. Easy as that. Okay, now, guys, RNA is very important to understand. It's the single-stranded molecule. It consists of a nucleotide, it's made up of nucleotides, subunits, but now it has 4 of the nitrogenous bases. They're the same as the ones from the DNA, but no, it does not have a thymine. It has got uracil. Simple as that. And then it plays a role in protein synthesis. Done. Now, what is important, last but not least, I want us to look at the comparison between RNA and DNA, because this question, guys, there's no way that you won't be asked this question. This is a very important question in life sciences. They're just testing your mind, and they know that most students, they confuse DNA and RNA. So they want to see if you understand thoroughly what's the difference between the two, because they want to test your knowledge on this. Okay, so it's very important to know that there are some similarities, but also there are some differences when it comes to these two nucleotides. Okay, what is the same is that these two nucleic acids, they've got nitrogenous bases, adenine, cytosine, and guanine. That's their similarities. They've got similar 3 nitrogenous bases. Okay, but now there's a different one. For instance, for DNA, we have got thymine, but in RNA, we have got what? Uracil here. We've got uracil in case of thymine, sorry. But in DNA also, we have sugar, which is called the deoxyribose sugar or the deoxyribose. But in RNA, we've got ribose. You see, that's different. And then this one is double-stranded. It's a double-stranded molecule. This one is a single-stranded molecule. Easy as that. Then you grab your 6 points or 3 points. I don't know how they they allocate the marks, but it's very important to know the difference between these two. They are similar because they have the same three nitrogenous bases, but now for a thymine, DNA does not have, so the RNA does not have thymine, and uracil is the one that is present in RNA, and DNA does not have RNA. What I meant is that in the place of thymine in RNA, we found uracil. And then in the place of uracil in DNA, we find thymine. The sugar in DNA or deoxyribonucleic acid, we find deoxyribose sugar. But on the other part, on the other one, RNA, we find ribose. DNA, double-stranded molecule. RNA, the single-stranded molecule. That's it, guys, for today. Thank you so much for watching. I'm bringing you the next one on protein synthesis. Please don't forget to subscribe. Don't forget to share to your friends. And don't forget to tell your friends to share to their friends. And of course, comment down below. If you have questions, please do not hesitate to ask me on the comment section. Thank you so much for watching, and I really appreciate for the support that you guys are giving me. Thank you. Bye-bye.